Good afternoon, everybody. Welcome to this TMS seminar that today will be held by Francesco Taglieri. Francesco is a student and is preparing his thesis for get his master degree in chemistry. He works in the field of synergistic catalysis that combine the properties of organocatalysts and photocatalysts, in defining the so-called domino reaction. Today he will talk about artificial metalloenzymes toward nature and beyond. So enzymes and transition metals there are many examples in nature of enzymes and transition metals, and Francesco will explain us what they can do together, let's say. Francesco, when you want, you can start.
Okay. Can I share the screen?
Yes.
Okay. Okay. So good afternoon, everybody, and thanks for introducing me, Professor Giansanti. I'm very excited to be here. Anyway, today we're going to talk about artificial metalloenzymes toward the nature and beyond. We're going to take a look at what artificial metalloenzymes are. We're going to see also some generation strategies to obtain artificial metalloenzymes. And at the end, we're going to see a really interesting application. So we're going to understand why this particular class of catalysts can be defined beyond nature. But let's start with defining what artificial metalloenzymes are, which is shortened as ARMS. They are a new class of synthetic biocatalysts, and they are formed by one or more metallic centers within a protein scaffold. The scaffold must be formed by at least 50 amino acidic residues and must be inactive when it is not bound with the metallic center. But enzymes and metallic ions are two sets that already intersect in nature, so natural metalloenzymes already exist. So which are the requirements that a metalloenzyme must respect to be defined artificial? The requirements, you must respect one of these three requirements, and all of these three requirements are connected with a generation strategy that we're going to see later. So the requirements are: the first, catalyzing a non-natural reaction; the second is to contain a new-to-nature protein scaffold; and the third one is to contain one or more non-canonical metal centers, for example, transition metals like iridium or ruthenium and stuff like that. So metalloenzymes, and specifically artificial metalloenzymes, combine the catalysis characteristics of both enzymes and metallic centers. From the first one, we have the higher reaction speed and turnover numbers, and also excellent selectivity and specificity. And they also can catalyze well-known natural reactions in mild conditions. The main property that we take from metallic centers is that they can catalyze reactions with no natural counterparts. So now let's skip to the generation strategies, how artificial metalloenzymes can be obtained. The first strategy is the reproposing of natural metalloenzymes, and this is based on the catalytic promiscuity that many of them show. In this case, promiscuity, it is not a bad word. and it is the ability to catalyze additional reactions to those of the main role. This refers to the possibility of both catalyzing the transformation of a substrate of different nature and also using different reaction mechanism and or different catalytic sites. So you choose some enzymes that are your candidates, and we all know that enzymes usually work in aqueous media. So you can change the medium, so you can change the solvent, the pH condition, or the ionic force of the medium, and also you have to change the substrate that the enzyme has to transform. So you do this pre-screening, and if there is one of these candidates that you have a promiscuous enzyme. The second strategy that we're going to see is the redesign or design. It depends on which point of view. And it relies on proteins of which it is possible to obtain a new scaffold, and this is called the de novo design, or you can obtain, or you can modify an already existing scaffold, and this is called redesign. The first one starts from computational studies, so you hypothesize your synthetic protein fold, and then passing from computational to synthetical studies, you have a new designed enzyme. The second approach starts from an existing protein fold, and by computational studies, you can hypothesize which amino acidic residues you can modify or substitute. But anyway, what you obtain is always a designed enzyme. The third strategy is the anchoring of artificial cofactors. This is used to provide the proteins with an activity that they wouldn't have. So non-canonical metal complexes with catalytic properties are introduced as artificial cofactors through appropriate anchoring pathways. So you have a protein that shows an anchoring site, and you also have a metal with some ligands, and those ligands bear a spacer or an anchor. So by conjugation of these two starting materials, you obtain a biohybrid catalyst. The anchoring pathways are three, and they are the electrostatic one, which uses electrostatic interaction to form your artificial metalloenzymes. These electrostatic interactions are really strong, but really dependent from the ionic force of the medium and also to the pH. So it is strong, but really dependent. It really depends on these factors. The second pathway is the covalent anchoring pathway, and it is based on the covalent bond between the protein and the ligand of the metal. Also, this interaction is really strong, but may inactivate your enzyme. So you have to see if it's worth it using a covalent bonding. The last pathway is the non-covalent or supramolecular anchoring pathway, and it uses the weak interactions between protein and ligands of the metal, and these interactions are really dependent from the dissociation constant of proteins and ligands. So it may be really strong or it may be weak. So by reconstitution, so you change the metallic cofactor, or you can add a new metallic cofactor, you can obtain a cyborg enzyme, which is a really cool thing. But anyway, now we're going to see why these metalloenzymes can be defined toward the nature and beyond. Now we change to a more chemical point of view, and we say that there are some reactions like olefin metathesis that are not catalyzed by the normal enzymes. Olefin metathesis is this strange thing that is showed in this picture. And to make it easy, is a simple pi bonding rearrangement, and it can be intermolecular, like in this case, or it can be intramolecular, and you can obtain a bigger closed cycle, or you can obtain a polymer. It depends on the condition of the reaction. Anyway, those reactions are not catalyzed by the natural enzymes or proteins, but it is possible to bind enzymes and proteins to organometallic cofactors that can do it. In this way, the synthetic and employment perspective are broadened. Those are some of the organometallic cofactors that are used to conduce this reaction. They are called Grubbs catalyst or Grubbs-Hoveyda catalyst. It depends on the generation that you're taking. And you have not to remember all the structures, but the main things that you must know are the metallic center, that is a ruthenium metal. It is not a common ion that you may find in natural enzyme. And also the ligand. This particular type of ligand is really strongly bound to the ruthenium and can be easily functionalized, as we're going to see later. So these new systems have potential for a bioorthogonal chemistry. With this term, these terms concern with the chemical reactions that may occur into living systems without interfering with the natural metabolic reactions. So if these systems have potential for this type of chemistry, why not to insert them into a living reaction medium such as bacterium? There's a little problem. Okay, moment. Okay, but the implementation of artificial metalloenzymes into living systems poses some difficult problem to solve. For example, the first one is the scaffold expression. So you have to be sure that your proteic scaffold is expressed by the bacteria. If it is not, you have to introduce it by the outside, or you have to genetically change your bacteria. The second problem to afford is the cofactor uptake. So you have to introduce from the outside to the inside the cofactor. And you have to be sure that it is not destroyed when it is taken up by the bacteria. The third problem to afford is the artificial metalloenzyme assembly, and it could be spontaneous or reactive. In the first case, we are lucky because the complex spontaneously form. But if it is reactive, you have to change some condition, like for example temperature, or you have to change the medium, or for example you have to add some other reagents, and so you have to to check a lot of factors. The last two problems are the cytotoxicity of the product and also the inhibition of the metalloenzyme. The first problem, the cytotoxicity, is referred to the product because when you introduce your substrate into the bacterium, or it can be obtained by a precursor that is inside the bacteria, you could obtain a product that is cytotoxic. So you're gonna kill your bacteria, and this is not what we want. So you have to be sure that the product and also the substrate are not cytotoxic for the bacteria. The last problem, the inhibition, may occur when there are some species, like in this case is showed the glutathione, and this species can inactivate your metalloenzymes because they really strongly interact with metal centers. So if they interact with the metal centers, it will be not used to catalyze your reaction. But even if there are a lot of problems when you have artificial metalloenzymes into living beings, in 2060 the research group of Yeshek and Ward could implement a streptavidin-based artificial metalloenzyme linked to a functionalized metathesis catalyst into coli bacteria. One moment. It's not my fault. My PC is not so good. Okay. So streptavidin is this protein and is a homotetrameric receptor protein with a really high affinity for vitamin B8. Vitamin B8 is also called biotin, and as you can see, we have a dissociation constant that is really, really low. So they really strongly bind with each other. And the functionalized metathesis catalyst is this type of catalyst. So we have a Grubbs-Hoveyda catalyst with a ruthenium center and this ligand. And as I said before, this ligand can be easily functionalized. And in this case, the ligand has been functionalized with biotin. So So the type of anchoring that they choose to use is the non-covalent anchoring pathway. So once they were sure that the streptavidin was expressed by the bacteria, they were also sure that this cofactor was easily taken up by the bacteria, because biotin has a really high affinity for the streptavidin. So these two were not a problem in this case. And the reaction that they decided to conduce is this metathesis of this product, that it is not cytotoxic, and also the product isn't toxic, because this is umbelliferone, that is a fluorescent molecule, so you can also follow the reaction rate by fluorescence measurement. And there were no inactivation by glutathione or species like that, because they were sure that there wasn't a species like that in the periplasmic space. So they conduced 3 types of experiments that are showed in this table. And the first is referred to a complex between streptavidin and biotin ruthenium with some amino acidic residues that have been changed. So the whole complex is indicated as biot-ru sub-mut. The second experiment is with the natural streptavidin and biotinylated ruthenium. And the last one is conducted with the simple ruthenium biotin. Those curves are referred to the kinetics of the reactions. And in this table, we have some really interesting parameters that now we're going to see. So we have, for the first, we're going to see the Vmax, that is the asymptote of the curve, and is the max velocity that the reaction can reach when the substrate concentration tends to infinity. So the higher is it, the better will be for the reaction. And as you can see, in the first experiment with the changed streptavidin, we have a really high value of Vmax. As a second parameter, we're going to see the Michaelis constant, is the concentration of substrate. The concentration of substrate when we are at the half of max velocity. And as you can see, in the last experiment, they obtained the lowest value. A lower value indicates a strong interaction between the substrate and also the catalyst, and this is not so good. So when you have high value, it is better, because there are not strong interaction between the substrate and the enzyme. The last is the ratio between the catalytic constant and the Michaelis constant, and this is a form to indicate the efficiency of the reaction. And as you can see, in the first experiment, we obtain the highest value of this parameter, and this is so good because it indicates that the changed streptavidin with the biotinylated ruthenium works real better instead of the other 2 experiments. So to conclude, artificial metalloenzymes are among the latest arrivals in the scientific landscape as a junction point between the artifice and the natural. They are the beginning of a change in their possible branch of uses. For example, they can be used in microbiological branches or, for example, pharmacological. The scientific progress represented by studies like these is the proof that the challenges posed by the nature are difficult, but also that knowledge can take them beyond the ladder. So, thanks for your kind attention, and that's all, folks.
So, thank you, Francesco, for your for this seminar. Very interesting. This is, as you said, a very new research field. Many interesting. I think it offers many opportunities.
Can I stop? One thing. Can I stop sharing?
Yes, okay. You can.
Okay.
Now the discussion is open. If someone wants to ask a curiosity or want to ask something, you can write on the chat. So Marco has a question for you. Can you think of any industrial application that are currently being used to exploit artificial metalloenzymes?
Industrial application. It's a really good question. I think that, for example, if you have a sort of culture of bacteria, you can, for example, give them the substrate as a component of the culture ambient of your bacteria. So you can give them the substrate, and then you can easily separate the bacteria from the substrate that you from the product that you wanna obtain. I really have not found a lot of things about artificial metalloenzymes. They are a really new catalyst class. So for industrial application, it is a little bit. It's too soon to think about artificial industrial application, but it's a really good question. I hope I answered your question.
Yes, I think so. Thank you for your answer. Are there any other questions from the audience? Don't be shy. So you talk about when you showed. I have a curiosity. When you showed us the slide with the explanation of the artificial metalloenzymes that can work in vivo, you showed that also eventual cofactor and substrate must enter into the cells.
Can I share the screen so I can take the slide?
But it's not necessary. It was just a curiosity.
Can you repeat the question?
No, it was just an introduction. So I was wondering when you want to that this artificial metalloenzyme work in vivo inside the cells. Mm-hmm. You showed us that the enzymes must be inside the cell, but also the cofactor, the substrate, have to enter into the cell.
Yeah.
So this is not trivial, I imagine, because sometimes, especially if you have a polar substrate, you need something that brings the substrate inside the cell, or you have to use substrate that can cross the cellular barrier, the membrane. It's not so. Maybe this is a limitation, or there are cases in which these are very recent fields. There are cases in which they use some delivery system to bring the substrate inside the cell or the cofactor inside the cell. Because if the enzyme works inside the bacterial cell, you have to bring this component, this reagent inside the cell.
This is really interesting, but maybe you have to study if you use, for example, a carrier for your substrate that you want to convert into a product. You you should be sure that your carrier will break and let the substrate be free in a specific compartment of the cell of the cell. So you have to study the kinetics of this phenomena, but it could be a really good way to introduce the
Because it's clear that we have to use someone like a transporter more than like a receptor, because here the delivery system has to must hallow the substrate across the barrier and then have to release it. So more a transporter than a receptor. It was just a curiosity. There is another curiosity over in the chat, just a curiosity. What is the best strategy? To modify the Metalloenzymes or to design new ones? Maybe I missed it.
You didn't miss it because I didn't say it, but I think that there is best way to obtain a metalloenzyme, an artificial metalloenzyme. Maybe it depends on which stuff you have to do your artificial metalloenzyme, and maybe you have also to study the primary, secondary, and so on structures of your enzyme, and you have to study how they're made, so you can decide if the enzyme can be obtained by modifications or if you can design a new one. Probably designing a new protein scaffold is really difficult because you have to synthesize by de novo a whole protein scaffold, and this is not an easy thing. But I think that there's no best way. It depends on which is the case. Did I answer your question?
I think so. I think you did. There are any other questions? As Michael writes on the chat, there are a lot of people today that are following this seminar. You don't have curiosity, or I was wondering when you talk about the fact that it must be careful, pay attention to the fact that the product that is formed by the reaction is not cytotoxic.
Mm-hmm.
But otherwise you may kill the cells. And I was wondering if we cannot think about to these systems as a drug to kill, I don't know, cancer cell or a bacterial cell that cause a pathology, a bacterial infection. So to use a reaction that is cytotoxic, is it possible to envisage an application like this? To use this approach, this artificial metalloenzyme as a drug that are internalized by the cells, and then when the substrate is introduced in the cell, they kill the bacterial cells that cause the infection. So.
Okay, so you're saying that we could use a pathogenic bacteria to.
Yes.
Okay, it's
Something like
really interesting.
to use a collateral effect and a problem as a solution.
Really interesting. Maybe it could be a future application. Why not? Maybe there are some problems when you have to do this, for example, on human beings. If you have a bacterial infection, maybe it is not the best way to introduce artificial metalloenzymes into human beings. But if
it depends on the nature of the bacterial infection.
Yeah,
maybe something on the skin. I don't know. It's just before arriving to the human beings, a lot of steps. I don't know. It was just a curiosity, an idea.
A really interesting idea. I mean, I think.
Someone else wants to ask something about this new approach? No? So, If there are no other questions, I thank again Francesco for his seminar, and I thank you all of you for your attention. And we will see you next Thursday for the TMS seminar. Good afternoon to everybody.
Okay, before you go, just 1 minute.
Sorry.
Just so like last week we have the form for multidisciplinary interest, which I'm going to put in the chat right now. So please answer the poll. It only has a couple of questions. It takes 1 minute to fill it up. It just regards the multidisciplinarity of the seminar. It means, does a person from another field understand what has been said in this TMS or not? That's it. So, and while we are filling up the form, I can update you on next week's TMS. We have Dr. Alessandra Biancolillo, who is a doctor in chemistry, and she will talk about chemiometry, which is an obscure science which no one understands. No, I'm just kidding. But next week we will understand more about it. So please be here. Also, if you are not chemists, because it's very interesting for biologists. In fact, we will be talking also about sarcopenia. So it's very interesting for everyone. Okay, so thank you everyone for being here today. Again, a very high very long attendance list, 135. So good job, Francesco and Luisa. And I'll see you all next week. Thank you.
Thank you, everybody.
Bye-bye.
Bye-bye. Good afternoon.